three types of food we eat all the time, right? We have carb, we have proteins, and we have fat. And the food we eat, what happens to it is that the food is oxidized. Okay, after we eat it, the food is oxidized. And we know in catabolism, right, the food is broken down into smaller components. And what do we produce in the process of catabolism? We produce ATP and we produce something else, few other small molecules. What are those molecules?
Like NADPH and FADH2.
Right, those are the electron carriers. Something else we also make. Think about the food. What happens to the carbon?
CO2 and ammonia.
Right? CO2, ammonia, and water, right? So that's what happens. So in the process, we basically eat very complex food, and it is broken down into the smaller components. And this process is like the food is being oxidized. Okay, that's what happens. Food is being oxidized after we eat it. And when something is being oxidized, what must also happen in parallel?
Something else is reduced.
Something else must be reduced, right? So when the food is being oxidized, the food we eat, electrons are leaving the food, okay? And those electrons are picked up by electron carriers, okay? So generally we eat food and this is being oxidized and the process is giving up electrons. And these electrons are then picked up by electron carriers. So we have NAD plus, we have NADP plus, and we have FAD. Okay, so these are the electron carriers. They pick up these electrons. And so NAD plus will go to NADH. NADP plus will go to NADPH. And FAD will go to FADH2. Right, so these are oxidized cofactors. Okay, that is why they can take the electron. And then once they have the electron, then they become reduced. So these are reduced. Right, so these, yeah, the food we ate, like, you know, we have breakfast, almost time for lunch. So after you ate food, the food was processed, okay, and is being oxidized. And the electrons are being picked up by this the oxidized cofactors, okay, and they are being reduced. Then these, so basically these are electron carriers. All they are doing is they are carrying electrons. And we know that electrons need a medium to be carried from one place to another, right? So in our body, these molecules are the carriers of electrons. Okay, they will carry electrons from one place to another. And by place, I mean from one protein to another protein. Okay, there are proteins which will take these electrons and do its thing. So, or enzymes rather. Then these electrons, they are going to our electron transport chain, which is inside the mitochondria. Okay, And inside the mitochondria there are membrane proteins. So we have learned a lot about transmembrane proteins last semester. So there are a lot of this in the inner mitochondrial membrane. There are a lot of these transmembrane proteins. There are 4 of them at least, which will take these electrons. There are 4 complexes, which will take these electrons from these electron carriers and just keep passing from one complex to another complex. Okay, complex 1, 2, 3, 4. And the last complex is complex 4. Then what it does is that this electron has to go somewhere, right? It has to be given to something. And what do you think it is given to? So all the food comes down to electrons. That is the bottom line from this semester. Just keep that in mind, that everything comes down to electrons. Okay, all the food, everything we eat, everything comes down to electrons. That's the sole purpose, is to generate electrons. And these electrons are picked up by these proteins in the membrane of the mitochondria. And what is the final fate of electron? So something must be reduced, right? Electrons are picked up by something, and that is being reduced. So can you guess what that is?
Is it oxygen?
So it is oxygen. And what happens to oxygen? After you reduce oxygen, what do you make?
Water.
All right. How many electrons do you need to reduce one molecule of oxygen to water completely?
4. 4.
4. Okay. So we have oxygen and we're going to water. And what is the oxidation state here on oxygen? 0. Here is -2. Right? So you need at least 2 electrons. But you have 2 oxygen, so you need 2 times 2. So that's 4 electrons. So we got to give 4 electrons. But because there are 2 oxygen, right? So to balance it, I need to put a 2 here. But that means we need 4 protons. Right? So 4 electrons, 4 protons. So that is what happened. The oxygen, like we breathe oxygen, right? That's what happens with oxygen. It goes to the complex 4, and there's an enzyme there. And that enzyme will take the oxygen, and with the help of the electrons from the food, it will reduce it to water. Okay. But there's something else very important, which is proton. Okay. We got these protons. So these protons, what happens is that those are being pumped. So just like pumping, you pump water, with pumps. Same thing here. Protons are being pumped across the membrane, again across the membrane. Okay, by the same complex, complex 4. Okay, and then what happens to those protons? Those protons are picked up by another enzyme called ATP synthase, and it is located on the other side of the membrane. So the protons that are pumped, basically there's a protein which will take proton from one side of the membrane to the other side of the membrane. Okay, and on this other side there is another enzyme, ATP synthase, and so ATP will take the protons and it will make ATP from ADP, right? So this proton movement literally rotates ATP synthase. It just proteins are not actually static. They're dynamic. They're always moving. And exactly that's what happens in the membrane. The ATP synthase like rotates like that. It rotates one place to another, just rotates. And protons are coming into the enzyme, and it is combining with ADP to make ATP and combining with PI. So that's just, the proton movement gives the energy to the ATP synthase to move so that it can combine with PI, okay, to make ATP. It is just, amazing how things work. So that's the sole source of the proton, is to make the ATP synthase move, the complex, the enzyme moves. Literally, it moves like that. And there is experimental evidence that ATP synthase actually moves like that. Okay, it just moves and rotates at a time. Okay, pumps one proton, picks up, makes ATP. So bottom line is that oxygen, we need oxygen so that the food we eat, the electrons we generate from the food can be consumed by the oxygen to make water. And so that is respiration, right? And in that process, the protons are used to make ATP. So if there is no electron transport, if there is no food, we will not get water, and so we will not get proton, right? So food is related to proton. And if we don't pump protons, then we will not get ATP, right? So proton is not directly coming from food. It is very important to know. But the food is giving electrons, so it is only giving electrons. But the protons are being pumped by the enzyme, and that proton is being used to make ATP. Okay, so just one more time is that the food we eat gives electrons. That electron is being picked up by oxygen, and the oxygen being reduced to water. In the process, 4 protons are being pumped across the membrane, and those protons are picked up by ATP synthase to make ATP. Right, so oxygen and electron combined will make water, and then proton pumping in the process, which is what makes ATP. Right? Makes sense? So that's sort of a general theme. Now, we eat, there are some trigger signals. So after we ate, we are full, okay, and it is called fed state, fed and starved. There are 2 states. So it's going to be fed and starved. Okay, so right after breakfast we are full, and so we are fed. Then the signal that the body gets is that, okay, I have food, I need to find a way to store it. First make ATP, and then the excess food we're going to store. And no matter what type of food you eat, carb or protein or fat, everything goes to fat. Okay. So the food we eat after the fed state, we make a lot of glucose in the blood, and that glucose has to be picked up by cells. Okay, and that is triggered by insulin. So insulin is a hormone that will trigger uptake of glucose from the blood into the cells. And this deficiency has, we know diabetes, diabetic patients have to take insulin shots. And there are 2 types of diabetes. We'll talk about that as well as we go along. But generally, insulin is the signal of storage. Okay, whenever we have food in the blood like glucose, insulin is going to trigger uptake of the glucose from the blood into the cells, so that the glucose can go on to glycolysis and go on to make ATP. Okay. Now, when we are starved, and extra carb is stored in as glycogen in the liver, we have glycogen. It is stored as glycogen, and it can serve as a short-term food. Like, after about 8 hours after meal, we start breaking down glycogen, the liver glycogen. That's the easiest access the body has to get ATP when we're not eating. So about 8 hours generally. And so the liver glycogen is the source that the body goes to first. Okay. And the glycogen will be broken down to get ATP. So that's what happens in the starved state. When we're starving, almost lunchtime. So you'll be eating lunch, and now another hormone has already might have started triggering that we need food. Okay, we need to get more energy, more ATP, and so that hormone is glucagon. So fed state we have insulin, and in starved state, glucagon. These are the two main hormones, and metabolism is under hormonal control by these two hormones. Okay, so glucagon is the signal when we're starving, and it gives the signal that hey, I need to find some source of ATP. Give me some source. So the body will go tap on glycogen, as I said, first in the liver, and to get ATP. And during prolonged starvation, then we need to, then we tap on our fat source and protein, like muscle breakdown. So prolonged starvation is bad because it can cause a thing called ketoacidosis. And so here, what happens is that we're going to make a lot of ketone bodies like acetone, acidosis. Yeah, we make acetone and some other things. And so these will release protons, and that will lower the blood pH, which is really bad. Okay, so prolonged starvation, when the body taps onto fat, then fat breakdown makes this small molecules like acetone and some other things, which releases protons. So the protons go into the blood, and it will lower the blood pH, which is really bad. And doctors do this test, is the breath test. Okay, when someone is just to know if someone has ketoacidosis set in or not, because acetone has a smell like nail polish remover, or if you work in organic lab, you know that the smell of acetone. So and it comes through the breath. So the doctors will first do the breath test to see if the ketoacidosis has set in or not. But you don't want to go there. That's bad. I'm not a dietitian. I'm just saying from the chemistry perspective. All right, so that's the general theme, and then extra food that we have, stored as glycogen or fat. Okay, those are the anabolic processes, or build muscle. Okay, from the protein we build muscle proteins. So that's the sort of the general theme. Let's take a break for a couple of minutes, then we're going to continue. All right, let's keep going. So yeah, we have metabolism. So there are two parts already we know. One is the anabolism. Just to give you a general overview, and so these is under hormonal control, and that is insulin. So insulin is the hormone that controls it. And here we have glucagon. There are some others, but these are the main ones, okay? Let me write another one here is epinephrine. Okay, so these are catabolic hormones. So we got anabolism, we got catabolism. So those are catabolic hormones. So basically, you know, the food we eat, the general, so we eat carb, right? We also eat protein, and we eat fat. So as I said, everything is going to be revolving around these three types of food, okay, in this semester. So the carb, you know, generally we make glucose, and the glucose then by glycolysis it goes to pyruvate, and from pyruvate it is a one-way street to acetyl CoA. So glycolysis itself is reversible. Okay, that's why this step, so this is glycolysis, and it has a both-way arrow, meaning it is reversible. So from pyruvate you can go back to glucose. Okay, but once we have pyruvate, then and after you make acetyl CoA, then from acetyl CoA you cannot go back to glucose, right? So this is one-way street. And whenever you make acetyl CoA, then you must enter the next cycle, which is the citric acid cycle. Okay. Tell me how many carbons are in glucose?
6.
So 6 carbon. What about pyruvate? How many carbons there?
3.
So it is split symmetrically, and that is why it is called lysis. We are breaking down glucose. So we got 3 carbons, 2 molecules of pyruvate we're going to get from one cycle of glycolysis. And what about acetyl CoA? How many carbons in acetyl CoA?
2.
So we'll get 2 of acetyl CoA, 2 carbon. So we have lost 1 carbon, right? What do you think might have happened to that?
Does it leave through CO2?
It does leave as CO2, right? We know that all the carbon we excrete as CO2. And so from pyruvate to acetyl CoA, we need actually, and it's an oxygen dependent process, okay, and we make CO2. Then, so what about the electrons? I have been talking that the electrons, we're going to get electrons from the process. So the electrons are here. From glycolysis, we get We produce 2 ATP directly, and we also produce 2 of NADH. All right, there we go. So we make electrons. And this is from glucose, we're going to make glycolysis, and that process will make 2 ATP and 2 NADH. This process here also makes NADH 1. And once we have the acetyl CoA, it is a 2 carbon, and the first thing happens to it is that it will produce citrate. In fact, so I'm just giving an overview, and citric acid cycle is one chapter on its own. Glycolysis is one chapter on its own. Okay, but I just want to give the sort of the flavor of the overview. Okay, so citrate, and citrate is actually 6 carbon. Then there are a lot of things here happens. A lot of things happen here. A lot of steps. And there is another thing here. It is called oxaloacetate, or OA. So OA is a 4 carbon. So a 4 carbon combines with acetyl CoA. So 4 plus 2, 6. You get 6 carbon citrate. Okay. But there are a lot of things here in between. So, there's another step here where we're going to get another CO2. Details we're going to look at later. We get, there's a step we get, ATP, but there are 2 of them, so we get 2 ATP directly. We get some NADH, and we also get another thing, which is FADH2. Okay, another electron carrier. We also get another NADH. All right, so all these steps we're going to get, generating all the NADH. And so all this NADH, from here, these, that, these, that, all of these guys are going to this electron transport chain, okay, which is in the inner membrane of the mitochondria. And they are these proteins I was talking about. They will just hand in the electron from one protein to another. And from there, all these electrons will make ATP by ATP synthesis. So that's what happens, okay? Everything comes down to electrons. And that's how food, the carbohydrate made ATP by this process. And so this is CAC, or citric acid cycle. Right. So important thing to notice here is that once you have acetyl CoA, okay, once you have acetyl CoA, then it cannot go back to pyruvate, so it must enter CAC. But if you have pyruvate, you can go back to glucose. So that's really catch here, is that pyruvate to acetyl CoA is irreversible. And when we have irreversible steps, then the delta G is always hexagonic. Okay, so wherever there is irreversible step, where it is going to be an hexagonic process, so the delta G is going to be negative. And this is how the body sort of regulates which process to continue and which not. Okay, So wherever there is irreversible step, those are generally rate limiting steps in the regulation. So that's how regulation happens, is that irreversible steps are regulated very, very tightly. So there are a lot of small molecules which regulate these irreversible steps very tightly, because think about it, if you have acetyl CoA, then you must have CAC, right? It cannot go anywhere else. So there must be a valid reason to make acetyl CoA. Okay, that is why it is regulated very tightly at this step. And same thing is applicable to when you do glycolysis. You're going to see that some steps are reversible. They can go back and forth. Those are not regulated, because it doesn't matter if it goes this way or the other. It does matter, but it's not going to make live or death situation. But the irreversible states must be regulated very tightly, so that those can only happen if there is a need. If there is no need, then those do not happen, right? Because you can't go back. There's only one-way street. So this is actually a very important concept in this semester, is about regulation. And the steps which has the most delta G, most negative delta G, are the ones which are regulated most tightly, because there's a lot at stake. If you have a very highly hexagonic process, you must make sure that happens only when it is needed. Okay, so the body finds a way to regulate that step very tightly. Because in a chemical, metabolic path, there are a lot of steps. Some are reversible, some are not. Okay, and not everything is regulated, but the irreversible steps are the ones that are highly regulated, and those also become the rate limiting step. Yeah, so the rate limiting steps are hexagonic, and those are the irreversible steps. So yeah, we're going to look at more in chapter 15, but just get that idea that this is one-way street. And then same thing happens for protein. So when you have protein, then we go to then we make amino acids. Then the amine part, okay, it is excreted as urea. So the amine part has nothing to do with citric acid cycle or anything like that. Amine, we don't get ATP from amine. Okay, it's just gone, excreted as urea. But the carbon and oxygen part of the amino acids, those can enter either, it depends, depending on the type of amino acid. These can enter either in pyruvate or in acetyl CoA directly. Okay, and then they will do the same thing. They'll go to the CSE and do the same thing and get ATP this way. Okay, everything producing ATP at the end. Same thing for fatty acids. So fatty acid, our storage fat is what? TAG, right? Triacylglycerol. We have seen that last semester. So we have TAG, and the TAG has the glycerol, right? The glycerol backbone, and it also has the fatty acid, right? Go back, revisit TAG. So the glycerol can actually go to pyruvate and go back to glucose if needed. But fatty acid does not go to pyruvate. It only goes to acetyl CoA. So that is why fatty acid breakdown is also highly regulated, because when you are making acetyl CoA, it has nowhere else to go. It has to go to CSE. and make ATP this way. And so there is a basic level of, so that's what I'm saying, that the citric acid cycle is highly regulated, because acetyl-CoA, once you have that, it has to go through the CAC. Okay, so it is highly regulated, the citric acid cycle. Okay, yeah, so that was the catabolism. And then anabolism, let's say when you have amino acids, we need to store it. Okay, we're going to make muscle protein, for example. And the fatty acid will be stored as tag. If you have excess glucose, then we can make glycogen. Okay, also cell wall. Right? So these are anabolic processes. And whenever there is anabolic process, that means you need to spend energy. So the ATP you make in the catabolic process is used to the ATP hydrolysis more often is to couple anabolic processes to make that happen. Because if you need energy, ATP hydrolysis is giving that energy. So that is why energy coupling concept comes into play. Basically, if someone is not able to move up the hill, you help. Okay, you help by the ATP to move up the hill. Okay, that's the same concept of this energy coupling. We'll see that next class. Yeah, I think that's enough. That's actually about time for today. Right, so just keep in mind that the food we eat goes to electrons, gives electrons. Those electrons are picked up by the electron carriers and delivered to the complex as in mitochondria. And complex 4 will take those electrons, reduce oxygen to water. In the process, it will make ATP, energy, pump protons, and that protons are used to make ATP. And just get the concept of reversibility versus irreversibility, and the general theme that whatever food we eat, everything goes to ATP in this process. And the fatty acids cannot make glucose. That is a very important concept. Fatty acids can only make acetyl-CoA. All right. So I guess that's all for today. And have any questions, let me know. I'm going to be here for a few more minutes. I'll see you next class.
Thank you. Have a good day.
Yeah, you too.
Thank you.
See you.
Can you go up to the first page really quick? All right, thank you so much. All right, thank you.
Yeah.
Hello. Do we need to read anything before the next class?
Read the first chapter.
So chapter 1?
No, not chapter 1. It's chapter 13, I believe.
Yeah, thank you.
Yeah.